Genetics, Section 5, the structure of DNA. In this section, we'll start off with a bell ringer. We'll look at the key ideas, and then the outline for the section includes DNA, the genetic material, searching for the genetic material, the shape of DNA, the information in DNA, discovering DNA structure, and then we'll summarize the section. So your bell ringer, DNA is often compared to a ladder or a spiral staircase. Look at the image and answer the following questions. How is the structure of DNA similar to that of a ladder or spiral staircase? And then how is it different from that of a ladder or spiral staircase? So we're going to compare and contrast this with a spiral staircase. Key ideas. What is genetic material composed of? What experiments helped identify the role of DNA? What is the shape of a DNA molecule? And how is information organized in a DNA molecule? And finally, what scientific investigation led to the discovery of DNA structure? DNA, the genetic material. The instructions for inherited traits are called genes. Genes are small segments of DNA, and all of those DNA genes are actually located on the chromosomes. So remember, a chromosome is this big bunch of DNA that's all wrapped and coiled together. A gene is a very small section of that DNA that's found in the chromosome. DNA is the primary material that causes traits to be inherited from parents. It's what is passed from parent to offspring, and it gives characteristics to related groups of organisms. It is a very simple molecule. It's only composed of 4 subunits, 4 different nucleotides. Searching for the genetic material. There were 3 major experiments that led to the conclusion that DNA is the genetic material within cells. The experiments were performed by Griffith, Avery, and Hershey and Chase. Griffith worked with 2 related strains of bacteria. Both of those strains caused pneumonia in mice, and he discovered that when a harmless live bacteria was mixed with a heat-killed disease-causing bacteria, that when the injection occurred into mice, the mice ended up dying. So they took a harmless live one, so that bacteria was not anything that would hurt the mice. But then they also took a heat-killed disease-causing bacteria, so they knew it was dead, and they injected them both into mice at the same time. And they found that when they did that, all of the mice ended up dying. So Griffith came up with the results that information that had been inside of the dead bacteria, the bacteria that were harmful to the mice, must have somehow been picked up by the harmless bacteria. And he discovered this idea of transformation. And transformation is the idea where a foreign genetic material found in the environment can actually be added to the genotype of another species. His experiments led to the conclusion that genetic material could be transferred between cells. He wasn't exactly sure how that could happen, but based on his experiment, he felt that some way there had to have been genetic information from the dangerous bacteria that was picked up by the harmless bacteria.
Transformation occurs when a bacterium takes up DNA from its environment. The process usually begins when a donor cell is destroyed and its DNA is released into the environment. A recipient bacterium then takes up a strand of the donor DNA. This is accomplished by means of a DNA-binding protein on the recipient's cell membrane. Another protein then exchanges the donor DNA for a similar piece of DNA on the recipient's chromosome. When the transformation is complete, a new stretch of genetic information from the donor is incorporated into the recipient bacterium's chromosome. In biotechnology, transformation is used to transfer an engineered plasmid into bacteria. For example, when the human gene for insulin is inserted into a bacterium, the bacterium is able to produce insulin.
So this is an example where transformation is used to benefit humans. We've actually taken and we've allowed bacteria to take up that human gene for insulin, and then the bacteria produce insulin for us. This picture is a picture of Griffith's discovery. So in experiment 1, he injected a mouse with live R cells, and the result was the mouse ends up living. Okay, so R cells are not dangerous to the mouse. In experiment number 2, he injects the mouse with live S cells, and S cells kill the mouse. So he knows that S cells are harmful to the mice. In experiment number 3, he kills S cells with heat. So those cells that before, when they were alive, they were dangerous or harmful to the mouse, he kills them with heat, he injects it into the mouse, and the mouse ends up surviving. And then in experiment number 4, he kills the S cells with heat, and he mixes it with live R cells. So remember back in experiment 1, R cells did nothing to the mouse. So he mixes the two. He combines experiments number 1 and experiments number 3. And in this case, he injects the mouse with a mixture, and then the mouse still ends up dying. So his conclusion was there had to be a transformation of R cells that ended up killing the mouse. So the R cells had to have picked up some of the dangerous genetic material that caused the death in mice. So searching for the genetic material continued. Avery wanted to determine whether the transforming agent in Griffith's experiments was protein, RNA, or DNA. So at the time, nobody really even knew what the genetic material was, or what the material was that was being passed on that ended up killing the mouse. So Avery, he ended up using some enzymes to destroy each of the molecules in heat-killed bacteria. His experiments led to the conclusion that DNA had to have been responsible for the transformation in bacteria. So DNA, through Avery's experiments, ends up being that substance that we start looking at as the genetic material, the material that codes for certain protein production, that codes for physical traits. In this case, with Griffith's experiment, it would have to be the material that's passed on that caused the production of some type of harmful either protein or enzyme or something along those lines that ends up killing the mice. Hershey and Chase, they studied bacteriophage. A bacteriophage is a virus that actually attacks bacteria. It infects bacteria. So they studied these bacteriophage viruses, and they end up using radioactive isotopes. And with those radioactive isotopes, they basically dyed the DNA. And they wanted to see when the DNA from a bacteriophage was dyed, where did that DNA end up when they put them inside of an experiment that held a whole bunch of bacteria. So these bacteriophage would inject, in this case, their DNA into the bacteria, and the bacteria ended up dying in the long run. But they ended up proving that DNA, not proteins, was actually the genetic material in viruses that was being passed on and put into the bacteriophage.
The Hershey-Chase experiment showed that genetic material is comprised of DNA and not proteins. Viruses, called phages, which infect bacteria, were grown in a nutrient medium that contained radioactive sulfur, called S35. Sulfur is present in proteins, but not in DNA. A second batch of phages was grown in a nutrient medium that contained radioactive phosphorus, called P32. Phosphorus is present in DNA, but not in proteins. The bacteria E. coli was infected with both types of phages. The infected E. coli cells were mixed in a blender. The lighter viral parts remained in the top layer of the mixture. The heavier bacteria cells sunk to the bottom. The radioactively labeled sulfur remained only in the upper layer containing the virus's parts. This meant that the protein was not injected into the bacterial cells. In contrast, the radioactively labeled phosphorus was part of the lower level containing the bacterial cells. This suggested that the viral DNA had been injected into the bacteria. The new generation of viruses that was produced in these bacteria also contained radioactive DNA, further supporting the idea that DNA was the genetic material.
So Hershey and Chase experiment really laid the foundation for some of the later work with DNA. It ended up giving us some evidence that the genetic material that was passed from parent to offspring has to be DNA and not proteins or RNA or any other substance. So the shape of DNA. DNA is a molecule. It is shaped a lot like a spiral staircase. It's composed of two parallel strands of linked subunits. And that spiral shape of DNA is known as a double helix. Each strand of DNA is made up of linked subunits called nucleotides.
Look closely at the DNA of this chromosome. You can see that it's made of two strands that spiral about each other. This structure resembles a twisted ladder, doesn't it? It's called a double helix.
So this short video just shows, first of all, shows the DNA, shows the double helix of DNA, but also if we go back there at the very beginning, it shows the overall structure, overall shape of a chromosome. So remember that DNA winds and coils around itself to create chromosomes within the cell. And chromosomes are found inside of the nucleus. So a nucleotide is made up of three parts. It's made up of a phosphate group, a 5-carbon sugar group, and a nitrogen-containing base. Phosphate groups in the sugar molecules of nucleotides link together to form a backbone for the DNA strand. And the 5-carbon sugar in DNA is called deoxyribose, which is kind of how deoxyribonucleic acid, how the name is created. And in the next slide, we'll look more specifically at each one of these subunits and what the overall structure of DNA looks like based on those subunits. So this figure, it is the model that Watson and Crick they ended up developing. It shows the double helix of DNA. So nucleotides are the subunits of nucleic acid. Each nucleotide consists of a sugar, a phosphate, and a nitrogenous base. So we can see here the phosphate, the sugar deoxyribose in DNA, and then a nitrogenous base. The different nitrogenous bases, adenine, guanine, thymine, and cytosine. And remember, adenine and thymine combine together. Guanine and cytosine will combine together. Guanine does not ever bind with thymine. Adenine does not ever bind with cytosine. And this is part of the reason why when copies of our DNA are made, we don't end up with as many mistakes. It would be a pretty bad thing if we ended up with copies of cells that had different DNA in it. We would end up with genetic mutations and abnormalities in proteins that were produced. So there's weak hydrogen bonds that occur between the nitrogenous bases. So these weak bonds allow for the easy copy of DNA later on. And then the sugar phosphate bonds, they make the backbone of each DNA strand. So sugar and phosphate alternate down the backbone, and that creates the bonds, or creates the kind of the backbone of the DNA strand that allows it to be held together fairly well.
Let's take a closer look at DNA. Here you can see that each strand of a DNA molecule is a long chain of subunits. These subunits are called nucleotides. This close-up diagram shows that a nucleotide is made up of three parts: a sugar, a phosphate group, and a nitrogen base. In DNA, the sugar is deoxyribose.
The information in DNA. The information in DNA is contained in the order of the bases, while the base pairing structure allows the information to be copied. In DNA, each nucleotide has the same sugar group and phosphate group, but each nucleotide can have one of 4 nitrogenous bases. So that really is what gives us the variability between our DNA and anyone else's or other organisms' DNA. Just simply the variation between those nitrogenous bases. The 4 kinds of bases are adenine, guanine, thymine, and cytosine again. And bases adenine and guanine have a double ring structure and are classified as purines. Bases T and C have a single ring structure and are classified as pyrimidines. A purine on one strand of DNA molecule is always paired with a pyrimidine on the other strand. Specifically, adenine always will pair with thymine and guanine will always pair with cytosine. The base pairing rules are detected by the chemical structure of the bases, and the nitrogen bonds between bases keep the 2 strands of DNA together. And remember, those hydrogen bonds are very weak bonds, which later on will allow for replication of DNA to occur. Basically, an enzyme will jump onto the DNA strand, it will unzip it between those hydrogen bonds, which leads to easy copying.
Notice that there are only 2 combinations of nitrogen bases that make up the base pairs in DNA. Adenine always pairs with thymine, and guanine always pairs with cytosine. This pattern is described by the base pairing rules.
So paired bases are said to be complementary because they kind of fit together like jigsaw puzzles. They fit together very nicely. Because of base pairing rules, the sequence of bases is known for one strand of DNA. Then a sequence of bases for the complementary strand can be quickly identified. And again, this is very important when we're making copies of our DNA. As more sections of DNA are added to the copied DNA strands, there's not as many mistakes that can happen. It's just like a jigsaw puzzle. Only one piece will fit in the place that it's supposed to go, which makes it a lot easier for DNA to be copied correctly. So discovering DNA structure, these 2 individuals, Watson and Crick, they were the information gatherers. They didn't really perform the experiments that led to the discovery of the structure of DNA. They basically just gained a whole lot of information from other individuals. Specifically, Chargaff, Wilkins, and Franklin, all of them did experiments that allowed Watson and Crick to slowly develop the 3-dimensional structure of DNA. Chargaff, he showed that the amount of adenine always equaled the amount of thymine. So they were always equal. So they knew that when they put these together, probably adenine and thymine would work together in some way. And the amount of guanine always equaled the amount of cytosine. So they started to realize that probably these 2 substances were pairing in some way. Franklin and Wilkins, they developed X-ray diffraction. So basically they took pictures of DNA with X-rays, and based on the picture that was created from those X-rays, it allowed for this DNA strand to slowly be developed, this model to be developed. So again, Watson and Crick, yes, they are extremely important because they eventually were the individuals that developed the 3-dimensional model, but we also need to understand that there's other individuals that contributed to this. They took a lot of information from other people to develop this 3-dimensional model. So they used both Chargaff's data and the X-ray diffraction, and they created their three-dimensional model off of those things. Their model showed a spiral staircase in which two strands of nucleotides twisted around a central axis. And in summary, DNA is the primary material that causes inheritable characteristics in related groups of organisms. There are three major experiments that led to the conclusion that DNA is the genetic material in cells. These experiments were performed by Griffith, Avery, and Hershey and Chase. And then DNA molecule is shaped like a spiral staircase. It's composed of two parallel strands of linked subunits. The information in DNA is contained in the order of the bases, while the base pairing structure allows the information to be copied. And then finally, Watson and Crick used information from experiments by Chargaff, Wilkins, and Franklin to determine the three-dimensional structure of DNA. So this was definitely a group effort in creating this three-dimensional structure of DNA over the time it took to create it.